In this lecture, we will discuss about hypothesis testing research designs. Hypothesis testing research designs are those in which the researcher tests the hypothesis of relationship between two or more variables. So these studies require procedures that would not only decrease the bias and enhance reliability, but also facilitate deriving inferences about the causality. So generally experiments satisfy such requirements. So when we talk of hypothesis testing research design, we refer to the design of experiments. And in experimental design, there are three principles. The first one is the principle of replication. According to principle of replication, the experiment should be repeated more than once. It means each treatment is applied in many experimental units instead of one. Now, what is the benefit of this? Now, by doing so, statistical accuracy of the experiments is increased. The result is more reliable. And you can see if we are repeating the experiment several times, we will get better results. Now, if we want to examine the strength of the concrete, then what we do, we make at least 3 samples for each mix. Then we take the average of that. So whatever is the effect of extraneous variables or any other effect or experimental error, that is reduced in this way. So generally in our codes, if you will see the IS codes, if you will refer them, then there are guidelines regarding the number of samples tested for each mix. Now, depending upon your code and what parameter we are going to test, you have to refer the code. And again, if there is precision required of particular level and you are not getting the result, or there is variation of more than percentage specified in the code, you have to repeat the test. After that, second is the principle of randomization. This provides protection when we conduct an experiment against the effect of extraneous factors by randomization. So here we should design or plan the experiment in such a way that the variations caused by extraneous factors can all be combined under one heading of chance. Now, for example, again, if we want to study the effect of temperature on concrete properties, the extraneous parameters would be that aggregates have come from different sources. The moisture content of aggregate of different lots may be different, or the testing may have been done in different seasons. So all these things, these may affect the strength, but we are not studying their effect. So these are all extraneous factors. So we should not make all the samples of the particular mix at one time or from one lot of the aggregates. We should randomize the resources, and then we should randomly select the sample from each lot and then check its property and take the average. Third is the principle of local control. So it is again important principle of experiment design. Here what we do, the extraneous factor or the known source of variability is made to vary deliberately over a wide range as necessary. This needs to be done in such a way that variability it causes can be measured and hence eliminated from the experimental error. This means that we should plan the experiment in a manner that we can perform a two-way analysis of variance in which the total variability of the data is divided into different components attributed to treatments. If we discuss the previous example, then to apply the principle of local control, we will test the effect of different aggregates as well as of the seasons on the properties of concrete. So we will have more than 2 variables in this study. So instead of two-way analysis, we have to go for multi-variant analysis. So in brief, we can say through the principle of local control, we can eliminate the variability due to extraneous factors from the experimental error. Experimental designs. So experimental design means the framework or structure of an experiment, and as such we are having many experimental designs. So broadly we can classify them in two categories. First one is the informal experimental designs and second one is the formal experimental designs. Informal experimental designs are those designs that normally use a less sophisticated form of analysis based on differences in magnitudes, whereas formal experimental designs offer relatively more control and use precise statistical procedures for analysis. So, the important informal experimental designs are before and after without control design, after only with control design, before and after with control design. And the various formal experimental designs are completely randomized design, randomized block design, Latin square design and factorial design. Now we will discuss all these one by one. The first comes before and after without control design. In such a design, a single test group or area is selected and the dependent variable is measured before the introduction of the treatment. The treatment is then given and then again the dependent variable is measured. The effect of treatment would be equal to the level of phenomena after the treatment minus the level of the phenomena before the treatment. Now, for example, what I want to find is the effect of construction of flyover on the number of accidents on a highway. We can carry out the survey before the construction of flyover and then after the construction of flyover. So then by comparing the results, we can comment on the effectiveness of constructing the flyover. So first of all, the level of phenomena means here we will check the number of accidents. Treatment is we have constructed the flyover and then we will check the level of phenomena, that is we will check the number of accidents again and then we will say what is the treatment effect, that is y minus x. Here this is the only way we can do, but sometimes there are the problems in which the, for example, time is involved. Now for example, I am now going to test the quality of water in Yamuna. I am testing it today. Okay. Now I am taking some measures to improve the quantity. Those measures may be any. We are adding something to the water or we are reducing the effluents from the industries into the water or so. It may be any measure to improve the quality. After 3 months, I am again going to take the sample and check the quality of the water. Now by checking the quality I will get the result in different parameters. What is the variation? But now time is involved there, 3 months. In this many extraneous factors can be there which I have not pre-assumed or determined or I am uncertain about them. Now for example, now lockdown is there. So what is going to happen in these 3 months? The pure water quality may improve. Maybe there are lesser effluents from the industries in the water due to the lockdown. So here I will not be able to assess the effect of my treatment. I don't know that whatever results I have got, they are just due to the my efforts, my treatment, or there are some other factors. So in that case, to remove these extraneous variables, we need to do the second type of option. The second type of design is before and after without control design. So here we are having two groups, okay, and the treatment is introduced in the test area only. The dependent variable is then measured in both the areas at the same time. Treatment impact is assessed by subtracting the value of the dependent variable in the control area from its value in the test area. So the basic assumption in such a design is that the two areas are identical with respect to their behavior towards the phenomenon considered. If this assumption is not true, there is the possibility of extraneous variation entering into the treatment effect. So here the data can be collected without the introduction of problems with the passage of time. So in this respect, this design is superior to before and after without control design. Now, in this case, if I want to check the purity of water, then what I will do, I will take the one sample which I will keep like this only without any treatment. I want to know the BOD, for example. So I have to check it after 5 days. So what I will do, I will take that one sample of water of that sample, I will keep it like this for 5 days, and one sample I will add the treatment and then I will keep it for 5 days, and then I will check the BOD in both the samples, and the difference will give me the effect of the treatment. And then the third one is the before and after with control design. So in this design, two areas are selected and the dependent variable is measured in both the areas for an identical time period before the treatment. The treatment is then introduced into the test area only, and the dependent variable is measured in both for an identical time period after the introduction of the treatment. So the treatment effect is determined by subtracting the change in the dependent variable in the control area from the change in the dependent variable in test area. So this is superior to the earlier two designs, because here it avoids the extraneous variation resulting both from the passage of time and from non-comparability of the test and control areas. So in this case, you can see that what we will do, we will keep a sample, we will take a sample from any sewage effluent, okay. That sample we will study the BOD before the treatment. So here we will consider the level of phenomena without treatment. Then we will take one another sample in which we will introduce the treatment. Then we will study the level of phenomena after treatment and level of phenomena again in the untreated sample without treatment. So your net effect, that would be y minus x minus z minus a. So here we have two samples before treatment. In one sample we have put the treatment and then we have checked its value, and the second sample is left like this for the same time period, and then we have checked the level of phenomena without treatment. So here we are having at the same time period what is the difference in these two, or without treatment effect is also subtracted to get the net effect of the treatment without extraneous variable of the time. Then we come to the formal designs. In formal designs, the first one is completely randomized design. This design is based on two principles: principle of replication and second is principle of randomization. So in this, what we do now, for example, we want to assess the experiment, we want to do the experiment, but we want to apply the principle of replication as well as the principle of randomization on that, so that the extraneous factors, they are removed. The experimental error is reduced. So here we are having two group simple randomized design and random replication design. First, we will consider the two group simple randomized design. So in two group simple randomized design, a sample is selected randomly from the given population and is randomly assigned to the experiment and control groups. The experimental and control groups of such a design are given different treatments of the independent variable. But the limitation of it is that it does not control the extraneous variable, and as such the result of the experiment may not present the correct picture. So here you can see that as I discussed earlier, that the extraneous factors are the moisture content of different lots of aggregates. So while testing, for example, if I will make all the samples with one type of treatment from one lot and the another type of treatment from the another lot, then what will happen? There may be effect of the type of aggregates, because in both we are using different type of aggregates, same type of aggregates but with different conditions. So extraneous variable is there. To remove this, what we can do, we can make few samples of each treatment from each lot, and then we can randomly select the specimens and then we can check. So here the population is our total specimens that we have made from different lots of the aggregates. Then we have randomly selected the sample and then we have randomly assigned them different treatments, or we can say we have randomly taken for control group and experimental group. And then the different treatments, for example, I want the different treatments of the admixture or cementitious supplements. So in one I have added maybe fly ash, in another I have added methacholine. Then I have given them treatments. Or I can say I have added 10% fly ash in one and the 0% fly ash in another, or maybe 20% fly ash in another. So here the extraneous factor due to the different properties of the different lots of aggregates, that has been removed. But in this case, what was there? We have randomly taken the samples and then randomly assigned the experimental groups. But what about the treatments? Now, as I told you that I have taken the treatments as the fly ash. And again, the fly ash that has come from the different sources. First, when I started, I got the fly ash from one source. Then for another batch, I got it from another source. Then there may be difference of properties in the fly ash. So that again will create another extraneous variable. To remove that, we are having two group simple random replication design. So in this design, what we do, the effect of differences in treatment, these are minimized by providing a number of repetitions for each treatment, and each repetition is called a replication. So there are two populations in the replication design. The sample is taken randomly from the population available for study and is randomly assigned to 4 experimental and 4 control groups. Similarly, sample is taken randomly from the population available to conduct the experiments and then it is randomly assigned. So here you will see that we have taken the sample randomly, we have assigned the aggregates to all the samples randomly, and then the treatments also, they are randomly selected from different sources. We have added for all the samples, then they are randomly applied. So the treatment is also randomly applied and the experimental groups or sample has also been taken randomly. And then we have made number of samples and then we have taken average value. So the replication is also there. So this is a random replication design. After that comes the randomized block design. So in randomized block design, the principle of local control can be applied along with other two principles of experimental design. Here the subjects are first divided into the groups known as blocks, and then within each group the subjects are relatively homogeneous in respect to some selected variable. So in this, third principle is what? Principle of local control. In this, we have to control the extraneous variable. It means what we do, we randomize the treatment also, and then we treat them with extraneous variables also. We then calculate their effect also. So here one more factor, that is extraneous variable, that is also taken as one variable, one dependent variable. So its effect is also calculated. So it is analyzed by two-way analysis of variance. Then comes the randomized block design. After that is the Latin square design. So it is generally used in agricultural research. Now, for example, an experiment has to be made through which the effects of the 5 different varieties of fertilizers on the yield of a certain crop is to be judged. In such a case, the varying fertility of the soil in different blocks in which the experiment has to be performed must be taken into consideration. Otherwise, the results obtained may not be very dependable. Now we want to judge the effect of the fertilizer. But if the soil is also changing in the different fields, there is different type of soil or its properties are different, then this will become the extraneous variable because it will also affect the fertility of the soil. Similarly, if we are using the seeds, there may be impact of different types of seeds on the yield. So to overcome such difficulties, the Latin square design is used when there are two major extraneous factors such as the varying soil fertility as well as the varying seeds. So here you can see that both the effects, they are measured. Here each fertilizer appears 5 times but is used only once in each row. So the field, it is divided into the rows and columns. So the different types of seeds we have taken, different fertility levels we have mentioned. So for one particular type of seed, it is put in all the fertility levels. Another type of seed, it is also put in all the type of fertility levels. Or you can say that the particular fertility level in this all types of seeds, they are put by making the rows and columns. So here in each row we are having one type of fertilizer in one block only. So the treatments in Latin square design are so allocated among the plots that no treatment occurs more than once in any one row or any one column. So the two blocking factors, they are represented through the rows and columns. So the advantage of this experimental design is that it enables differences in fertility gradients in the field to be eliminated in comparison to the effects of different varieties of fertilizers on the yield of the crop. But there is again one limitation. What is that? Along the row or along the column. Okay, we have there may be, in this way also there may be variation in the fertility level. So it is that although each row and each column represents equally all fertilizer varieties, there may be considerable difference in the row and column means both up and across the field. Then comes the factorial designs. Factorial designs are used in experiments where the effect of varying more than one factor are to be determined. Factorial designs are of two types. First one is the simple factorial design and second one is the complex factorial designs. Now in the previous designs we have just studied the effect of one independent variable on the dependent variable. But when there are more than one independent variables, then we have to go for the factorial designs. First is the simple factorial design. Here the effects of varying two factors on the dependent variable are considered. Simple factorial designs may either be 2 by 2 simple factorial design or 3 by 4 or 5 by 3 or any type of simple factorial design may be there. So there are two treatments, okay, of the experimental variables and two levels of the control variable. As such, you will see there are 1, 2, 3, 4 cells into which the sample is divided. Each of the four combination would provide one treatment or experimental condition. Subjects are assigned at random to each treatment in the same manner as in a randomized group design. Then the mean of a different cell may be obtained by taking the means for rows and columns. So here you can see, now for example, I have taken the two, here you can see that two treatments and two control variables are there. So the two treatments, two are concrete, are, let us say, in one sample, in one type of mix we are adding fly ash, and in another type of mix we are adding methacholine, okay, and the control is the property. So here for example I want to take the property of compressive strength as well as the flexural strength. So in this way the table would be made and we will get the values for the compressive strength for fly ash mix also, for methacholine also, flexural strength for fly ash also, for methacholine also. So two treatments are there and their effect is calculated. The other thing here can also be that my treatment is, in one I am adding 10% fly ash, in another I am adding 20% fly ash, and I am calculating the compressive strength for first source of fly ash and second source of fly ash. Okay, so the fly ash is from different sources. So here in this way I can find the compressive strength for first source for 10%, for second source, and then I can find out the mean, which will remove the extraneous factor of the sources. So here we can find out the column means also. In similar way we can find out the row means also to get some values or some interpretation of the results. Similarly, when we are using the more treatments, if 4 treatments are using, then it would be 4 by number of controls 3. So here one example is for 4 by 3, in which 4 treatments I have given: fly ash, methacholine, 5% fly ash plus 10% methacholine, and 10% fly ash plus 5% methacholine. And then I am measuring 3 properties: compressive strength, flexural strength, and tensile strength. So this is a 4 by 3. So similarly it may be 5 by 2, it may be 5 by 4, or any type of factorial design. So here if I change the treatments, what I am doing, I am using the cementitious compound of fly ash only, with 5% fly ash, 10%, 15%, and 20%. Similarly here my control is only the compressive strength, and but the fly ash is from different sources, source 1, source 2 and source 3. Then by adding all this and taking the mean, I can have just the effect of adding 5% fly ash without considering the effect of the source, and so on. After that comes the complex factorial design. A design which considers 3 or more independent variables simultaneously is called a complex factorial design. In case of 3 factors with one experimental variable having 2 treatments and 2 control variables, each one of which having 2 levels, the design used will be termed as 2 by 2 by 2 complex factorial design, which will contain a total of 8 cells. So here you can see, first of all, I will explain these. Now, for example, my experimental variables were, first, I was considering the fly ash and methacholine. But again, in fly ash samples, I have 2 types of samples. One is with fibers and second is without fibers. Similarly, in methacholine, I am having 2 types of treatments, that is with fibers and without fibers. And for all those samples, I want to know the compressive strength and flexural strength. So in this way, I will have treatment. Then each treatment, here you will see that each treatment, it is having 2 levels. Okay. And similarly, the treatment B, it is also having these 2 levels. Second is, for each treatment or each level, we want to know these 2 properties. So 2 properties for one, 2 for other, 2 for other, 2 for other. We will have 8 cells. So this is a complex factorial design. So here you will see that we are having, if from this complex problem we can infer the results for simple factorial designs also. Now here it is possible to determine the main effects for 3 variables, that is 1 experimental and 2 control variables. We can also determine the interactions between each possible pair of variables, and these are called first order interactions. And interaction between variable taken in triplets, these are called second order interactions. So if I am talking of the first order interaction, it means what? I am considering this part only. So here, this is the complex factorial design. So if I am considering only this part, 2 treatments are there and 2 controls are there. So here it is just a 2 by 2 simple factorial design. Or if I am considering, not considering this thing, then again 2 treatments are there and 2, again this is a 2 by 2 simple factorial design. But if I am considering every variable, then it is 2 by 2 by 2 complex factorial design. Similarly, if next we will take the example of, this is the first level interaction, and then similarly you can have any level of interaction from this, and it can be generalized to any number and combination of experimental and control independent variables. The greater the number of independent variables included in a complex factorial design, the higher order of interaction analysis is possible. So we can say that there are several research designs, and the researcher must decide in advance of collection and analysis of data as to which design would prove to be more appropriate for his research project. Thank you.